We're going to be talking today about the project that's been mentioned a couple of times on trying to optimize blueberry fruit nutritional quality by selective stress of the plants. And we're going to go through to the next slide. We're going to split this talk into two parts. One I'll give, which is the first part, in which we're looking at the effects of the light distribution under the covers. There you've got the three covers that we have at Arimba, in which the experiments were conducted. Most of the experiments were conducted. And then Gareema will look at the output, the effects of this that we've had on the fruits that we've been growing. So I'll go through, and what I want to do is look at the dye, and what the aim of this part of the project was to work out why it was in the literature that we wanted to make sure that what we got the benefits of the leaf of the fluorescent dyes, and to see why it was in the literature that some people claim are getting very good increases in growth, and other times it doesn't seem to be so good. There seems to be an inconsistency in the results that people are getting. So what we've done, well, I'll just give you a better background. Here's the absorption spectrum of the polycarbonate with the leaf 660. You can see how the light is absorbed in this region, and then it's emitted in the other. Now, what's going to become important is that degree of overlap between those two spectrum, which I'll get onto in a second. As you can see, this is one of our curves that we've got from our growth chain, from the polytunnels, where the yellow line represents the clear polythene, which is the middle of the three polytunnels we had, and this is the effect that it's having. The red light you can see is increased in that region, and using the purple film, it's going down into the far red. So here you can see an example of what Chris showed, where you get an advantage, a growing advantage, because you're increasing the amount of red light over what's getting through in a normal polytunnel. Okay, now we wanted to see we wanted to see what kind of conditions was they going to give us this advantage. So, and the first indication there's a little bit of an issue with this comes from some careful experiments that Gareema conducted, measuring the light spectrum over a day. Now, they are quite difficult to collect, especially in the last 2 years since we've had all this rain. Planning to get a set of data over a day and in three polytunnels, moving the things around, means you don't always get consistent results. It's a problem of measurement using using a single instrument and moving them between the polytunnels. You do get some change. But even here, we can start to see that in the region 300, 400, we do get an advantage from this, from the film, which we were interested in because the blue spectrum is what we would expect to trigger the production of flavonoids. There is a small increase, but it's not a great deal, although it looks large in percentages. We're comparing it with a clear polythene film, which would wipe out nearly all UV light. So we're getting a small advantage because the parylene dye does emit light in the UV region. So we were hoping that might be significant. We'll talk about it, like Gareema will talk about whether it did give us an advantage. So that's the reason for this lot. But you can see that even here, there is an advantage. The period of the day in which we're getting an advantage in that way that we're increasing the red light in a clear day is confined mostly to the mornings and maybe to the late afternoon. We get much better results if we do this on a cloudy day. So our cloudy day results seem to give us a greater advantage over those we get over a clear day. Now, in order to understand that, we decided to have a look in more detail at what was happening and at the published models for the behavior of the parylene dyes in polycarbonates or other plastics. So here is what I've called the fate of the photons. And the fate of these photons, if you've got incident sunlight coming in, of course, you could get scattered loss from the surface, depending on how rough it is. You may get reflected light. But what we're really interested in the light is the light that entered the film. So a light photon, if it's in the spectrum of green or yellow, may hit a red particle, and at that point it would be absorbed. You'll get electron transition, and then a relaxation, and then an emission of a lower particle, and that's the red Stokes shift that Chris described earlier. Now, what's important is that that particle could go off in any direction. It's a function, it's not true scattering, because there's a chemical process involved in here. It's not an instantaneous process. There is, even though it's very quick, there is a small delay in the emission of that particle, and out it will come. Now, if it comes off at an angle less than the critical angle, which is the gray area on this graph, then it will exit. If it doesn't, if it's greater than that angle, it will be absorbed. It may undergo total reflection, and it may indeed reach a photovoltaic cell at the end, but it's far more likely that that photon will then hit another red particle, and since that red molecule is not now receiving light, which is red, it will just absorb that as light, and that's the cause. That's caused in that overlap region between the emission and the excitation spectrum, which I showed earlier. So overcoming that comes down to things like geometry, which is what Chris was describing. So if we keep that in mind, and then imagine what happens when there's more than one photon. I can't put it on the diagram here because we get too cluttered, but we would get more of them, of these particles, are being hit by sunlight. And then what happens when all of the light is being hit at the within the space of the excitation and decay series? Well, we began to think that maybe there was a limit to how much light would actually get shifted by a given concentration of dye. Although that's not in the current models, it began to dawn on us that there was something here that what happened to light in excess of what the film could cope with? Was there a limit to the amount of light that could be spread? Now, we couldn't get that data in the field. It's really difficult to collect, as I've mentioned. Getting a consistent set of data over a day is quite difficult, but getting it, doing it this kind of experiment, we had to go to a more controlled system. There's just too much variation in sunlight over a day. So what we did, we took that indoors and we put it into a system like this, where we are focusing the beam of light and we're passing it through the film, which we put onto an integrating sphere. Now, if you remember back a couple of slides ago, I said that the thing of when a photon of light hits a molecule, it will be scattered in all directions. So what this apparatus enables us to do is separate out the light which is hitting dye and emitted and being scattered, and that will be collected in the sphere, and the light which is going straight through. And the way we measure that is by putting a white cap on here and collecting it all and doing it by difference. So we can work out what light is going straight through, and we can change the intensity of the light that's going into the system. So we could control the conditions, and we could isolate the light scattered by the dye. Now, what was gratifying in a way is from our hypothesis that there was light that was going straight through unaffected because it was over the saturation, is that at all normal wavelengths of light that you're putting into it, you get the same amount of emission, the same amount of area is absorbed at all intensities of light that go through. So no matter what intensity you're putting in, down to a minimum, which I'll get to later, you will get the same amount of scattering, and the remainder of the light is going straight through unaffected, as our hypothesis suggested. So we've got an amount of light that's shifted is constant, and it's dependent on its history. And Chris was talking about the diffusive power of the films. Yes, there is. It does have a diffusive power. It's a set amount at a given light intensity. All right. So what did we find from our project? Well, so far, we do have circumstances in which we will get, this is data that we have collected from our lamp. The light is shifted, and you do get an increase over polycarbonate, over clear polythene, an increased amount of red. But that only occurs when the light drops below a critical level, and we get that advantage. The lower the intensity of the light, the bigger that advantage becomes. So if you can imagine on that graph that if you drop the yellow line downwards so that it was more of a curve downwards to the point where the red line touches the zero, and effectively that is the most efficient use of the polycarbonate film, is at the point where all of the light is finding a dye molecule and undergoing the transformation. As you increase the light from that point, increasing amounts of light are going straight through the film and through to the target unaltered. And at that point, what we start to find is that we do get a drop off, and we don't reach the level of the light that's going through the polythene. So in other words, the advantage of the fluorescent films diminishes as a product of the intensity of the incident radiation. And in bright sunshine, it doesn't produce an advantage, but it is pretty good at doing it when it's cloudy or reduced, or if you control the incident light downwards, so it's not full sunlight, and therefore you get the advantage of the red film over whatever you've got as your clear polythene control. So it suggested to us that in our experiments, we were going to see a seasonal variation. We would get an advantage in the winter months and probably get an increase in growth, and not get an advantage in terms of the red light during the summer months. And I'll pass over to Gareema now, who will describe what we did get.
Thanks, Richard. I will share my slide. Let me know if you can see my
Yeah, we can see that, Gareema. That's good.
Perfect. Thanks, Richard. So from the next few slides, we are going to talk about how these spectral quality and intensity from the covers enhances or impacts on the blueberry fruits and the leaves, all the growth parameters. So the increasing awareness of health benefits has provided impetus for healthy eating, which among other options has included a higher intake of fruits and vegetables. The incorporation of nutrient-rich fruits in the diet has provided immunity against major diseases and reduced the risk of acute and chronic ailments. In this study, we have utilized blueberry as a model crop, as it is an excellent example where the consumer demand has increased globally due to its higher bioactive compounds present in the fruits. Given these bioactive compounds are strongly influenced by environmental stresses, therefore, in this project, we aim to address how we can enhance further, given under the protected cropping covers, these bioactive compounds reduces. So the hypothesis and the target of this aim in this project was to address how we can further enhance the bioactives in the fruits through light and stress intervention. However, in this particular talk, I'm just going to touch upon the light stress, not the abiotic saline stress, which we have also studied in this project. So this is the overall overview of my entire PhD. So there were three major interventions. To start with, the first is determining the effect of light distribution and light intensity, that how it impacts on fruit quality and growth parameters. The second intervention was the use of blue LED light. As previously Richard mentioned, that in most of the protected cropping conditions, the blue light has been eliminated or filtered off. And for that reason, it is important to provide blue LED light to the crops as it increases the bioactive compounds in fruits. So in the second interventions of our study, we have supplemented blue light to the blueberry crops so that we can see how it impacts or enhances the bioactive compounds. And the third intervention of my PhD was application of saline stress under polytunnel conditions, so that we can see how it impacts fruit and overall growth rate. In this talk, I'm just going to focus on the first intervention, which is how light distribution and intensity is going to impact blueberry plant and fruit. So each wavelength has a unique effect on plants, ultimately influencing their growth of leaves or fruit quality, overall parameters of plant growth and development. The use of photo-shifting covers, which we did and conducted studies in this project, was the first L-leaf 620, which shifted light towards the red wavelength. So in this region from 600 to 740 nanometers. And the second photoselective covers, which we utilized, was shifting light towards far red region, which is 740 onwards. So the hypothesis of this study was when the plants will get exposed to these photoselected covers, it is going to grow. So the growth rate will increase in terms of plant height. And at the same time, it will enhance the fruit quality. That means bioactive compound concentrations. Also, we also had a second intervention where we also utilized the combination of blue and red and far red light, just to understand how this combination of wavelength works and impacts crops. So this is the data from the blueberry leaves, which we have seen the certain significant results. So just going through the chlorophyll content, the photoselective film, which shifted light towards red wavelength, which is 620 L-leaf, had the highest chlorophyll content compared to other treatments. The reason could be the production of more chlorophyll was required for plants to maximize the capture of available diffused light, which ultimately resulted in increase of the chlorophyll content in the photoselective films, getting more of the red wavelength. In terms of plant growth, which is plant height, treatment, the same one which was receiving the high red wavelength, has also increased in the plant height. The growth parameters of the second photoselective film, which was getting more of the red wavelength, it wasn't significantly different to other treatments. So overall, in terms of growth parameters, the study concluded that the leaf film, which was getting more of the red wavelength light, which is 620, that enhanced vegetative growth under red shifted light conditions. In theory, red light also known to promote photosynthesis and carbohydrate production, and likely that is the reason that the photoselective cover 620 facilitated growth responses. In terms of blueberry fruits, we have calculated bioactive content in the fruits, which were utilized through 4 of the key components. So the total phenolics, total flavonoids, anthocyanins, and antioxidants. Those were the 3 parameters for us based on which we have concluded. So in terms of phenolic, anthocyanin, and flavonoids, commercial clear plastic performed much better than any other covers. However, when we were talking about total antioxidant content in the blueberry fruits, photoselective covers, which shifted light towards far red light, that has increased the total antioxidant in blueberry fruits. The reason probably could be because of high shading, and the spectra has been shifted towards far red wavelength, which resulted in increase in the antioxidant content. However, for the antioxidant, flavonoid, and anthocyanin content, blueberries did not surpass that of the result when compared with the commercial plastic films, indicating that still there is an improvement which is required, for example, increasing the scattering of the light, transmittance rate, and probably thickness. In regards to photoselective film, which shifted light towards red, which is LD620, that has shown to have a trade-off in terms of it increased the growth parameters, but not significantly to the growth rate, fruit bioactive compounds. So, which is obvious, there must be some trade-off where the energy allocation will be going more towards growth rate rather than fruits. So the conclusion from the study is the light shifting covers improves blueberry growth rate, but some improvements will be required, and we have seen the photoselective covers performing the best during the shaded environment or during the cloudy and winter days. The commercial covers have also been increased in terms of bioactive compounds when compared to photoselective films, and probably these photoselective films can be further enhanced when making sure the modifications in the scattering, transmittance, some dye modifications, as Chris mentioned in his slides previously, that they're working on. So in conclusion, the future use of these photoselective covers could include the combination of red and far red or red and blue light that could target both growth of the plants and at the same time bioactive compounds of the plants as well. Thank you.